So today we're going to be talking about DNA and RNA structure. DNA is deoxyribonucleic acid, and it stores the genetic material for a cell inside the nucleus. RNA is ribonucleic acid, and it has many different roles within the cell. So firstly, there's mRNA, which stands for messenger RNA, and this is involved in transcription. Secondly, there's tRNA, which is transfer RNA, which is involved in translation. Both transcription and translation will be covered on later videos. So, nucleic acids are composed of recurring units called nucleotides, which are monomers. And nucleotides are made up of three main components. So firstly, we have a pentose carbon ring, which is either deoxyribose or ribose. So if you remember back to topic 2, carbohydrates, there's two different types of sugars that are pentose sugars that we need to know. There's deoxyribose and ribose. Let me just draw them real quick. So and if you remember, the difference between deoxyribose and ribose is, as the name suggests, the 3' carbon lacks a hydroxide group. Instead, it just has a hydrogen group. So it's actually missing an oxygen. So the second part of the nucleotide is a phosphate group. And the third one is a nitrogenous base. So we need to know how to draw a nucleotide, and the phosphate group is represented with a circle, and it's connected to the pentose sugar by the 5' carbon, so by this carbon. Then we have the pentose sugar to represent either deoxyribose or ribose, which is attached to a rectangle, which represents the nitrogenous base. And this is attached to the 1 carbon. So DNA and RNA are both polymers of nucleotides. However, there's many differences in their structures. The first one would be the number of strands present. So DNA is double-stranded. And you may have heard that it has like a double helix structure. We'll go over this in more detail later on. Whereas RNA is single-stranded. Second difference is obviously the type of sugar. So deoxyribose is for DNA, and ribose is in RNA. Just extend this a little bit. Then we have the base composition. So DNA is A, G, C, T, or adenine, guanine, cytosine, and thymine. In ribose, we have A, G, C, U. So adenine, guanine, and cytosine stay the same. However, thymine is replaced by uracil. So this is a diagram of a DNA molecule. And by the way, all of the diagrams that I've mentioned so far in the video we have to be able to know how to draw them for the exam. So in DNA, nucleotides are linked together into a single strand or a polynucleotide chain via condensation reactions. And these are anabolic reactions, meaning that they convert monomers into polymers. So again, the monomer in this situation is a single nucleotide, whereas the polymer is the whole polynucleotide chain. And again, DNA is a double helix, meaning it has two strands. So it has two polynucleotide chains, which are bonded together via hydrogen bonding. So the phosphate group attached to the 5' carbon of the sugar of one nucleotide binds to the hydroxyl OH group attached to the 3' carbon of another nucleotide. And this results in a phosphodiester or a covalent bond between the two nucleotides and the formation of a water molecule. So a phosphodiester bond would be this. So it's between the 3' OH group and the 5' carbon phosphate group. The two polynucleotide chains in the DNA, as I mentioned, are held together by hydrogen bonding between complementary nitrogenous bases. This means that adenine will always bind to thymine and guanine will always bind to cytosine. And this is because adenine and guanine are both purines, meaning that they're double-ringed structures, whereas thymine and cytosine are single-ringed. So if thymine were to bind with cytosine, there just wouldn't be enough space for a bond to properly form. However, if two purines were to bind, then it would just be too squashed together because they're both too big. So that's why we have this complementary system with A binding to T or uracil in RNA and G binding to C. So A and T, as you can see here, are binded together by 2 hydrogen bonds, whereas C and G are binded together by 3. A nice way I like to remember it, it's very scientific, is that adenine cheats on thymine with uracil and RNA, which is why their relationship is weaker, so they only have 2 hydrogen bonds, whereas guanine and cytosine, they're very strong, so they have 3 hydrogen bonds. So in order for the bases to be facing each other and thus being able to pair via complementary base pairing, the strands must be running in opposite directions. And this is called antiparallelism, or just antiparallel. And as you can see here, this is running in the 3' to 5' direction, whereas here, this is running in the 3' to 5' direction. So yeah, as you can see, they're running in opposite directions, which is why it's called antiparallel.